I would like to continue this session where I would like to present my work on the construction of a out of equilibrium metabolic network in both a nanometer and micrometer-sized vesicle. So basically, in this presentation, I'm following up on the work that Bert and Meteor already have presented. Um, so what we are doing in our lab is we're trying to construct a synthetic cell using the bottom-up approach. And what we have done is to take the JCVI Syn3A cell as a sort of a benchmark to compare a potential bottom-up synthetic cell with the top-down minimal cell. And what we have done is to plot the genome sizes of several microorganisms, including the genome of the JCVI, versus their cell volume. And what you can see is the genome of the JCVI, so approximately 0.5 megabase pairs, fits well within a volume of 0.03 cubic micron. And this volume
Jelmer,
is very similar. Yes.
Jelmer, I don't see your next slide. So it's
Oh, okay. So basically what I was trying to do is to explain this graph. So we plotted the genome sizes of several microorganisms versus their cell volume. And I said that the JHVI, its genome fits within a volume of 0.03 cubic micron. And this is very similar to the volume of a vesicle with the diameter of 0.4 micrometer, 400 nanometer, which is the type of vesicle that we are working on in our lab. Also, the JCVI has approximately a rough estimation of 500 genes. If we take into account the genes that encode, for example, for the membrane proteins, basically we can reduce this to about 250 soluble proteins. And what we did is we calculated the encapsulation probabilities of the soluble molecules inside our lipid vesicles in our system. And this is what you see, for example, in this graph over here on the left. If we increase the size of our system, so increase the size of our vesicles, then the probability that we'll have at least one of these soluble proteins on the inside increases. And of course, the probability of having none of these molecules inside decreases. Of course, one molecule of one soluble protein is not a lot. So we also plotted the case where we have at least 10 of these molecules, so 10 copy numbers of this protein on the inside. And then you can see that you need to increase the size a bit more. But this is for only one particular, so only one soluble molecule. We also looked at the case that is more relevant for the JCVI. If we would encapsulate 250 different types of molecules, so let's say 250 soluble proteins, with an abundance of at least 10, so 10 molecules of these 250 different types of proteins, then you can see that we would need at least a vesicle with a size of 130 nanometer, which is the radius. And this is well within the size that we typically use. What we also did is to take a look at the membrane proteome of the JCVI. And what we have done is that we plotted the protein surface occupancy versus the radius. And then the red area in this graph is the protein surface occupancy that is found in nature, in microorganisms. And the black line is the membrane proteome of the JCVI. So if we were to reconstitute the membrane proteome in JCVI in an occupancy found in nature, we would need a vesicle of at least 200 nanometers, a radius of at least 200 nanometers. What we also did is to convert this graph. So instead of plotting the surface occupancy, we plotted the lipid to protein mass ratio, basically how much lipids per protein we have. And then the blue area represents the part, represents our current technical possibilities. So this is what we can technically achieve in the lab. And basically, if we want to reconstitute the membrane proteome of the JCVI inside our vesicle system, we would need a vesicle of at least 800 nanometer of a radius. Well, of course, you also need some space for the peripheral membrane proteins and some free floating lipids. So in the end, we would need a vesicle of at least 1 micrometer. So what I would like to do is to work that I did on the ADI pathway, so the arginine breakdown pathway. It's the same pathway that Bert has already presented, consisting of 3 soluble proteins and a transporter. So the transporter imports arginine, which is then converted on the inside via 3 enzymatic steps into ornithine and ATP. The ornithine is again exported by the same enzyme, and in that way we can have an out of equilibrium metabolic pathway. And what I did is to have this system into 2 types of vesicles. We have a submicrometer vesicle, which is more in the size range of the JCVI cell. We call these vesicles large unilamellar vesicles. And what these allow us to do is to do ensemble measurements. So we can measure many of these vesicles at the same time. And what I also did is to have that system, so the arginine breakdown, in a micrometer-sized vesicle. These vesicles are typically more in the size range of a mammalian cell. And what this allows us to do is to do single cell measurements. So we can really observe the behavior of our arginine breakdown pathway in single vesicles. And how we make these GUVs, so this larger vesicles, is that we have a objective slide, a microscope slide, on top of which we then put a layer of gel. So this can be a polyphenol alcohol-based gel or an agarose-based gel. On top of that, we would add lipids and then make a sort of a sandwich out of this, in which we have 2 of these objective slides with a spacer in between. Then in the spacer, we add our soluble components. This can be our proteins, fluorescent probes, anything. And what happens then is that these soluble components can diffuse through the lipid bilayers and through the gel, forming these buds, which will grow over time and detach. And in that way, we have our GUVs. Then, of course, when we want to do some measurements on them, we need to trap them. And how we trap them is we have this microfluidic chip. This device was developed in the lab of Tom Robinson. And what this chip has is like several lanes, in which in each lane there is a trap-like structure, a bucket-like structure, which traps all the GUVs. So we can have a continuous flow of GUVs, which are collected in the buckets. And then afterwards, once we have the trapped GUVs, we can wash them, and we can also add our substrate solution. So we could add our solution containing the arginine. How this looks like in practice is that we have a cover slip with the chip on top of it. We have an inlet reservoir where we add our GUVs, where we can add our solutions. And we have the outlet where we would connect a needle and a syringe. And with that, we can have a continuous flow of liquid through the chip. And what we would then see under the microscope is one of these buckets filled with GUVs, and we can monitor this bucket with these GUVs over time and observe their behavior. So I did the ATP production via the arginine breakdown in 2 vesicle system. First, I would like to present the work on the smaller vesicles, so the vesicle sizes in the size range of the JCVI cell. What we can see when we add arginine, in the case of where we have a arginine-ornithine antiparter, we can see that the ratio of ATP over ADP increases massively. So we have the formation of ATP in this system. If we don't have the transporter present, which is represented by the lower graph, the line stays flat, so indicating the ratio of ATP-ADP does not change, and thus we don't have any ATP formation. So what we can conclude is that in these submicrometer-sized vesicles, we do need a transporter. But then what I did is to upscale the system. So I had the same system, but instead of doing it in submicrometer-sized vesicles, I had micrometer-sized vesicles. If we have the arginine-ornithine antiparter, we do see again an increase in the ATP over ADP ratio, indicating that also in a GUV, in a micrometer-sized vesicle, we have ATP formation. Now, when we have the same system, but without the antiparter, we still see the formation of ATP. So apparently we don't need the antiparter in our giant vesicles, in our mammalian cell-sized vesicles, to have ATP formation. Having the antiparter is important because ATP formation is quicker, it's faster, but it is not essential. So what we thought is that probably arginine somehow enters the GUV. And so what we did is we made our GUVs empty GUVs with only a sensor for cationic amino acids on the inside. And then we added our substrate, so arginine, but also ornithine and lysine. And what we saw is that upon the addition of arginine and all the other cationic amino acids, we saw that the fluorescent ratio increases, indicating that these amino acids are entering the GUV. So they are able to permeate the membrane, at least in our GUV setup. Of course, we also tested ATP. And when we add ATP, the line stays flat. So ATP is not permeable inside our GUVs. Now, this is all nice. We have a system that we can produce ATP. But of course, we would like to use the ATP that's being produced. And one of the things that I'm working on is a system for membrane growth. In our lab, we have two of these systems. One system was slightly presented by Bert before, is where we produce DOPA. So it's a very simple phospholipid from fatty acids and glycerol 3-phosphate in an ATP-dependent manner. You can see in this curve below that over time we have the formation of DOPA. And what I'm particularly working on is then the conversion of PA to PG. PG is another type of phospholipid, also in an ATP-dependent manner. In the system, CTP is required and is released later on as a CMP, which I can recycle back to CTP using two soluble enzymes in an ATP-dependent manner. Well, here I show that I have recycling of CMPs, so I see that CMP decreases over time and CTP is increasing. And over time, I see the formation of DOPG. So this is represented by this dark blue curve. Now, my ultimate goal would be to combine both of these pathways. So to have a small vesicle system in the size of the JCVI that produces ATP via the arginine breakdown pathway, and to encapsulate this in a bigger vesicle. So let's say having a sort of a mitochondrial system inside a GUV, in which I on the GUV would have the phospholipid synthesis pathway. And that's, of course, we also take advantage of the fact that arginine can permeate the GUV. And then we would need to use the ATP carrier as well to excrete the ATP, which can be used for PG formation. The challenge here, of course, is that we need to be able to encapsulate these LUVs inside GUVs, and that we would need to be able to efficiently produce GUVs, so the big vesicles with the membrane proteins. So yeah, that would be the ultimate goal. And with that, I came to the end of my presentation. I would like to thank a few people, especially Henry. Henry was involved in many of the permeability studies that we have done. Also, Michiel was involved there. And Arnold, Martin, Eleonora for their work on the lipid synthesis. And of course, Dirk and Bert for having me in their lab and their supervision. And with that, I would be happy to answer your questions.
So much to think about. So again, with our minimal cell, we have gotten rid, so relative to the organism from which it is based, wild type Mycoplasma mycoides, we have gotten rid of the vast majority of the membrane coding protein genes. Mm-hmm. Now, a lot of those are probably synthesized at relatively low levels, but it would still, I expect that the surface of the cell has a lot fewer membrane inserted proteins than the wild type organism. Whereas the data that you showed at the beginning of the talk makes me think that if it just has a relatively small number of proteins, then in order for the wild type protein molecule to exist, the cell would need to be a lot larger. Am I and it's not. Am I misinterpreting your explanation?
No, I think so. I think you're correct. So I think the right now the JHVI has membrane occupancy of around membrane protein of about 30%. Yeah, I would say in a wild type cell, it's very similar.
And how did you calculate that?
So we calculated this based on the well, on the data that is there on the eLife paper from Breuer,
Yes.
and then assuming that these membrane proteins are like cylindrical shaped in the membrane part. Yeah.
Got it.
And of course, it also depends a little bit what is the doubling time that you're taking into account. I think here we took the doubling time of 2 hours. If you decrease the doubling time, you need less proteins, and then of course the occupancy also lowers.
Okay. And as you may know, so in adaptive laboratory evolution experiments where the cells, remember the wild type organisms were never intentionally, because the people who identified these organisms and originally worked with them were fundamentally interested in pathogenesis. And so they tried to passage the cells as little as possible in laboratory media. But two groups that Kim and I were both associated with showed in 2023 that we can, by growing the cells continuously in the SP4 media, we can get about 15% faster growth relatively quickly. So that might still reduce the number of proteins that you needed on the surface, I guess.
Yeah, I guess.
So John, perhaps I could add something. We have extensively looked at your eLife paper, and I would say all the important membrane proteins are still in your SYN3A and 3 for pH homeostasis and so on. Also, amino acids like glutamate, you need a lot. Those systems are there. I don't remember about the peptide transport right now, but as Miguel already pointed out, in principle, you can also get 20 amino acids in by using a broad specificity tripeptide transport protein. I think some of that is also present. So I would say
there is an oligonucleotide, an oligopeptide transporter that is essential.
Yeah. And so basically, you can use 20 transporters for 20 different amino acids, but you can also use an oligopeptide transporter. And if that is present at sufficiently high levels, then that will supply the cell with all the amino acids. And I think that is what we are seeing in your JCVI, and that is what we are planning to do in our bottom-up constructed cells. And, and of course, we will encounter problems, but at some point, if we are in regular contact, we may be able to sort things out and help each other in that respect. So I would say potassium transport, pH homeostasis, amino acid transport, a couple of other important nutrients. It's still there. I wouldn't be surprised that it is relatively upregulated, these molecules, relative to the parental mycoplasma, but the parental mycoplasma will have additional systems that are no longer present anymore.
Yes.
This is speculation, but that is a fair suggestion I would like to make.
And I have always speculated that there may be one or more amino acids that cannot cross the membrane in any form other than as an oligopeptide.
Yeah.
But because otherwise, I would think that the in our SP4 media, which has large excesses of amino acids, the oligopeptide transporter is still essential.
Yeah, I think you're correct. Yeah, I would agree with that. Yeah.
We know it's not cysteine because you can make media that doesn't have the source of oligopeptides in it is a protein that rather than using tryptone, we use a plant-based product that doesn't have cysteine. So, but there's still 19 other candidates.
Yeah.